Good afternoon. Thank you for joining. My name is Lindsay Cookson, and I'm the Water and Science Program Coordinator for the Lake Champlain Committee. On behalf of the Lake Champlain Committee and Tetra Tech, we would like to welcome you to our first three-part series on three-part webinar series on benthic cyanobacteria. We are really excited to partner with Tetra Tech to bring together experts across the country to share about their research and experiences studying benthic cyanobacteria in lakes, rivers, and streams. While many people are familiar with floating cyanobacteria blooms that occur on the water's surface, benthic cyanobacteria are an emerging area of research that presents unique ecological challenges and as well as public health challenges. We hope that this series really does provide valuable information for volunteer monitors, lake associations, researchers, resource managers, and anyone interested in freshwater health. I would like to thank the Lake Champlain Basin Program as they've provided funding to make this webinar series possible. But before we get started, just a couple of housekeeping notes. The presentation will last about 30 minutes and will be followed up by a question and answer session. Please feel free to submit your questions throughout the presentation using the Zoom chat, which is that in the bottom middle button. We'll save them till the end, and then we'll try to get through as many as we can. We also ask that people keep their microphones muted throughout the presentation just to kind of reduce the amount of noise that's going on. And we are recording the webinar presentation today as well for those who are unable to attend live. So now it's my pleasure to introduce today's speaker, Gina LaLiberte. Gina serves as the statewide harmful algal bloom coordinator and inland beach monitoring coordinator for the Wisconsin Department of Natural Resources. Today, she'll be discussing identifying benthic cyanobacteria in lakes. And so thank you so much, Gina, for joining us, and the floor is yours.
All right, well, thank you. Let me get the presentation set up. How does that look to you? Seeing the main slide?
I can see it. Yep, I can see it.
Okay, great. And I will switch to a laser pointer too. There we go. Yay. Okay, well, thank you so much for having me. Really appreciate the opportunity to talk with you today. So something that's really important to recognize about cyanobacteria, otherwise known as blue-green algae, is that they live everywhere, but blooms are really not a problem everywhere. We're most familiar with things like these planktonic or the floating blooms, but benthic blooms are kind of all over the place too. This bottom center photo is benthic cyanobacteria, or benthic means bottom dwelling. Cyanobacteria living in a mud puddle in my neighborhood. Over here, these are some benthic mats that have floated to a surface in a stream in my neighborhood. And this top center photo is actually a terrestrial cyanobacteria. And really, unless you have a lot of nutrients present, you're not going to be able to fertilize the growth of cyanobacteria to really, really high levels. So you might not necessarily see a lot of benthic cyanobacteria growing in lakes, but they most certainly are there. And we'll talk about some of the things that might make them more prominent or more noticeable to you, unless you're, say, snorkeling in a lake. I want to point out some resources in addition to this webinar. The Interstate Technology and Regulatory Council a few years ago convened a work group, a national work group, to put together guidance for planktonic or the floating cyanobacteria and the benthic or bottom-dwelling cyanobacteria. And you could find that at these web addresses here on the bottom, hcb-1.itrcweb.org. And they have some really great identification resources there as well. One of those resources is a Learn to Identify Cyanobacteria Blooms video. It's about 30 some minutes long. It has this lovely, gentle guitar music, so you can feel soothed as you look at really significant blooms. But this was put together with help from the Lake Champlain Basin Program. So some of you might know or have worked with Angela Shambaugh, who is retired from the state of Vermont, and she was one of the leads on this project. And you will see her in this identification video. So that's a really useful resource as well to kind of help reinforce your cyanobacteria identification skills. So when it comes to cyanobacteria, um, benthic blooms or benthic cyanobacteria can be really kind of cryptic. So here on the left, we see this planktonic bloom. It's really thick. It's probably smelly. It's giving you some of that telltale blue coloration from the blue pigment that's released as those cells decompose and die. Over here on the right, all of these dark colored blobs that you see here, these are all benthic cyanobacterial mats that have floated to the surface. And they might not be really noticeable. You might think they're just something else, decaying plant material or something else. So that's really one of the big challenges around benthic cyanobacteria in lakes. They can be really cryptic. Here's another example of some more cryptic benthic cyanobacteria. This kind of looks like possibly decomposing filamentous green algae, but these are actually little mats of benthic cyanobacteria that have accumulated on the edge of this pond. And here's a real tricky guy. This is a little blob of benthic cyanobacteria. It's growing right next to this fresh filamentous green algae, so it's trying to sort of blending in with that. And then here is some older filamentous green algae that's covered with a lot of diatom epiphytes. So again, reinforcing that they can be really cryptic. So you do need to kind of train your eyes in what to look for in figuring out if something's benthic cyanobacteria or something else. And one of the things that's kind of counterintuitive about benthic cyanobacteria is they need really clear water and, in many cases, really good water quality to grow. We're used to most often planktonic blooms growing in water that has a lot of nutrients, maybe kind of moderate to poor water quality, just because there's so many nutrients in the water, and those nutrients are fueling the growth of planktonic blooms. But if you think like a benthic cyanobacterium, what is it you need? You are growing on the bottom of a lake. You photosynthesize, you've got to have access to sunlight in order to do photosynthesis. So you have to have that clear water, which is going to allow sunlight to reach you on the bottom. So here are some examples. Here, these are some benthic mats of cyanobacteria that probably floated up along with this filamentous green algae from the bottom of a lake, and you could see how clear that water is. Over here, all of these little kind of teal dots in the water, those are some, it's a species of Plantothrix that's really common in lakes, especially in northern Wisconsin. We kind of see these floating to the surface later in the summer in these clear water lakes again. So they are going to need clear water usually in order to grow because the sun has to reach them on the bottom of lakes. You probably recognize this guy, Gliotrichia, from your ID cards that the volunteers have. This is also sort of more of a planktonic species too, but we can include it in the benthics because Gliotrichia is really good at moving up and down in the water column. It actually goes down to the sediments to kind of scavenge phosphorus and then floats back up towards the surface so it can get more sunlight. And it looks like little kind of tan dots in the water. They're about pinhead sized. So here you see they're, I'd say, probably like a third to a half of the diameter of a sesame seed. So these guys are pretty small. They can make blooms that look like tan or khaki colored stuff floating on the surface of the water. And this is what it looks like under the microscope. I always describe it as looking like a koosh ball or a cat toy. So that's Gliotrichia. So when it comes to other benthic and other benthic cyanobacteria, I'm going to concentrate now on benthic filamentous cyanobacterial mats. For the most part, a lot of the benthic mats that grow on lake bottoms tend to be filamentous species. So this is what a really large accumulation looks like floating on the surface of a lake in Wisconsin. And I think benthic mats of cyanobacteria are really interesting. And this is one of the reasons why, what we call the phycosphere, basically all of the organisms that live within and around the mats, really closely associated with them, are called the phycosphere of those benthic mats, and they can be really diverse. So again, here's that benthic mat example that I showed you. And these are a couple of microscope pictures of what's in there. It's not just one species of stuff. There's a lot of things growing in there. So here on the left, we have a couple of different genera of cyanobacterial filaments. Over here on the right, you can see some more of those cyanobacterial filaments, but there's a lot of other things growing in there too. These things right here are purple sulfur bacteria. Here and here are a couple of species of euglenoid type algae. Up here, this is a diatom. This guy actually is modal and it can kind of glide through that mat. And this is just a portion of a green alga called Clusterium. It kind of looks like a big green banana growing in that mat. So sometimes if I get samples of benthic mats and I kind of get distracted by all of the other cool things that are growing in there. So they can be really, really diverse. And I think that makes them a really important part of lake ecosystems. You've got this tiny little diverse ecosystem just living on the bottom of lakes. And one of the things that's really interesting about benthic filamentous cyanobacterial mats is that a lot of the species are motile. They can move, and some of them tend to have sort of this gliding or trembling type motion. And this is really useful for them because if those mats get disturbed, they can just kind of crawl around the bottom and reform as mats. And I'll talk more about that in a minute. So that was just some of the kind of trembling and gliding motion that you just saw. And then some of them are really active. So here's one species and another species they're really booking it through those mats. So some of them move fast, some of them move slow, but that motility is one of the adaptations they have growing as mats on the bottom. They're able to move around and take advantage of different spots and sort of recoalesce as mats if they're disturbed. So unless you're, say, snorkeling or scuba diving, you wouldn't usually see those benthic mats on the bottom of the lake. They're just down there minding their own business, quietly photosynthesizing. But as they photosynthesize in that clear water, they're putting out oxygen. And then sometimes they put out so much oxygen that the oxygen sort of forms bubbles within the mats, and then that buoys them to the surface where they can become a lot more prominent, a lot more noticeable. So here are some examples of those benthic mats that have floated up off the bottoms of lakes and rivers. So again, these sort of dark colored mats, often they are sort of a dark color, but here is a species that's more purplish in color. All of that green stuff around it is duckweed. And I'll show another picture of this in a little bit. This is a Planktothrix mat. Again, this is one that's really commonly floating to the surface of clear, especially clear lakes in northern Wisconsin in late summer. And then here's another mat that has floated up off the bottom of a sort of a marley lake, and it has sort of a golden color on top because it has actually lifted up some of the sediments along with the mat, and there's a lot of diatoms in that sediment too, giving it a golden color. So the colors can be very different, but kind of floating stuff. I'll give you some cues for what to look for in these benthic floating mats. So you might have done some of these simple field tests before as part of your volunteer work in assessing whether you're seeing a blue-green algae or cyanobacterial bloom or not. So if you have green water or floating mats, how do you tell if those are cyanobacteria or something else? So there's some really simple field tests that you can do. So one of them is a jar test, and this is really useful for knowing if green water contains those planktonic or floating cyanobacteria or something else. So basically, you grab some green water in a jar, you shake up that jar, and you let it sit for a while. And since planktonic cyanobacteria are buoyant, they will usually float and form a little scum on the surface. What you're doing in this jar is you're basically mimicking conditions in a lake on a day when there's no wind whatsoever, and planktonic cyanobacteria can float to the surface instead of being sort of mixed back into the water by winds and waves. True algae usually are going to sink. This isn't absolutely foolproof, and you really need to put water under a microscope to know for sure what's in there. But if you know you see these tiny green particles and you know it's not something like duckweed or water meal, that will usually tell you if you've got planktonic or those floating cyanobacteria in there. But here is my pro tip for using the jar test for floating benthic mats. You can grab some of that mat in water, shake it up, and then I set the jar close to a window. We talked about motility of some of the species that form benthic mats. So by putting it close to a window, quite often those benthic mats will crawl over to the side of the jar that has a lot of sunlight. So again, here, this part of the jar where you see the sun that was facing the window, you can see that it looks a little bit fuzzy and green, and that's because those benthic cyanobacteria have crawled over to the side of the jar so they can take advantage of more sun. So that's one example of how you might use a jar test. Here's another one. This is another example of some crawlers. So I've got some filamentous green algae that fell to the bottom of that jar, and I think there's some strands in here, and some of these guys just kind of crawled right up the strands and made this weird looking assemblage. And there's a little bit of stuff floating. I think there was also some planktonic cyanobacteria in there, but all of these guys sort of crawling along this matrix of filamentous green algae are benthic cyanobacteria. This is what they look like under the microscope. This is some Planktothrix. So another thing that you can do if you see floating mats in a lake is what's called a stick test. Basically, grab a stick, put it under the mat, lift it up. If it coats a stick like paint, like this, or if those mats just fall apart into tiny little particles, that's probably cyanobacteria. If the algae drape off the stick or a rake or forceps like you see here and they look like green hair, that's probably going to be filamentous green algae. But there is one exception. You do have to look really closely at the color. Microcyra is a type of cyanobacterium. It tends to be really dark colored, like you see here. And for example, for contrast, here is filamentous green algae on the right. Here's a penny for scale. So look closely at that color because microcyra is going to be the exception. I'm going to show you some more pictures. So here's what that stick test looks like with microcyra in a lake near Madison, Wisconsin. So you can see it's draping off that stick, but it has that really dark color. So that's microcyra. And again, that's the exception to the stick test. This is an example of where I typically find it in lakes around Madison. It looks like dark masses of hair that grow entangled with aquatic vegetation. So here, here, and here. And then in Lake Erie and Lake St. Clair, there are the right conditions there for microcyra to roll around on the bottom and make this little compact balls like you see there. And that's actually quite an issue in Lake St. Clair. They're seeing so many of these accumulating near shore that they get these big muck deposits forming along the shores of the lake as those decompose. And then if you can look really closely or use a dissecting scope, um, these filaments can be several inches or several centimeters long. And then think about this. This is a cyanobacterium, so that's absolutely huge. You can see it with the naked eye. Sometimes you see what looks like little breaks in the filament. If you stick it under the microscope, you can see that it grows as filaments in a thick sheath. And these breaks that you're seeing in the filament here are actually corresponding to sort of separation between those filaments inside the sheath that you see there. And then sometimes you'll see what we call false branching, sort of you'll have one filament and another one will sort of grow alongside it in a false branch. And then here's one more picture. And the reason I'm really kind of emphasizing what microcyra looks like. So again, this kind of black hair, here's aquatic plants underneath. This is probably some decomposing filamentous algae, but look for that black hair. It can produce toxins in some conditions, and I really try to share with people in Wisconsin, like how to identify this, because we did have a dog death last year that was potentially related to ingestion of microcyra. This is a dog that kind of liked to dig around in the water, drank a lot of water, and became seriously ill and died. And we think that it was exposed to a chunk of this stuff, accidentally ingested it, and that microcyra happened to be producing toxins. So do learn what this looks like. I don't think people, for the most part, are going to want something that looks like that going to be in their mouth. Adults sure won't. I don't think kids will, but dogs don't care. If it's just kind of stuff floating in the water, they might snap at it like it's a game or a toy or something. So if you see this, please keep your dogs away because it could be dangerous for them if the dogs ingest it. So this is another really common mat former. This is Oscillatoria, and I'm using this sort of broadly because taxonomists like to split things into different groups. So sort of Oscillatoria and its relatives. Again, this just tends to make sort of these dark colored mats that don't look like a lot. So just really dark colored stuff that could be muck floating. For me, this is a creek that's near my house. And for me, this is a sign of spring. The creek water is really clear in the spring. The stuff's growing on the bottom. It starts photosynthesizing like crazy, makes a lot of oxygen that bubbles up in the mats, and they float to the surface and then float downstream. So I go out and walk my dog over there. I'm like, is it spring yet? Have we seen the oscillatoria mats floating down the stream yet? And for me, that's a sign of spring. Here's kind of a close-up of a mat. It's not really dark colored on top because it's brought up some of the sediments from that bottom, but sometimes you can see what looks like black hairs sticking out of the side of those film of those mats. And those are the individual filaments. And here's a better example. Here is a mat that's brought up a lot of sort of tan colored sediments from the bottom, but all of these kind of black hairy type things that you see sticking out of the edges of that mat, those are oscillatoria filaments in that oscillatoria mat. And I mentioned this one before. This is sort of that purplish looking mat. So these dark purple mats surrounded by green duckweed in this sort of isolated lagoon area. This is what it looks like under the microscope. Oscillatory and its relatives. If you have access to a microscope, the cells look like, or the filaments look like stacks of pennies. So each one of these little penny looking things is an individual cell, and they all stack up in these big filaments. And this is another one that if you look really closely, it's a really big species for Cyanobacterium. So these guys are like almost half an inch long. You could see them with the naked eye if you look really closely, and you can definitely see them if you look at them with a hand lens or a magnifying glass. So this is what the close-up of that sort of purplish Oscillatoria princeps looks like. And again, back to Planktothrix. There are a number of species of Planktothrix. Some have this sort of bright teal color. There are some species that are more red too, and they can form these mats that grow on the bottom of lakes with really clear water, and then they can float to the surface. Again, I would say the beginning of August is when I start to get reports of this in lakes in northern Wisconsin. So this is Planktothrix. And again, that was Planktothrix that I talked about earlier with kind of the crawlers and the jar test. This is one of those motile things. So if you pop it under a microscope, these guys are going to be gliding back and forth and sort of going all over the place. And if you kind of let them sit undisturbed in water, they will recoalesce into a little mat because they have the ability to move around. They're motile. And then finally, I'm going to end with benthic cyanobacterial colonies or globules. So here's a kind of a bigger picture of some of these globules sitting on the bottom of a lake in southern Wisconsin. And that previous picture, here it is again, that is a Phanatheses stagnina. This is a particular species of cyanobacteria, instead of growing in filaments, it kind of grows in little cells that are in sort of a gelatinous matrix. Some of them can have more of this teal type color that you see here. More often, they're sort of a darker olive green color, so kind of dark colored. And these are sort of a pretty soft colony. You can actually pinch through them with your finger. So if you find colonies that sort of look like this, darker olive green to teal color, and you can actually pinch through them with your finger without much effort, that's probably going to be a Phanatheses stagnina. And I wanted to bring up this look-alike. This is actually a colonial protozoan. It's not a cyanobacterium. It's called Ophrydium. And that also grows in kind of these globular colonies. Here are some in the palm of my hand. They can get to be pretty big, several inches across, but more often they're about 1 to 2 inches in diameter. I want to point out that colors. This is a nice leaf green color. And the green color in those colonies come from these symbiotic green algae that actually grow within the individual Ophrydium cells. So together, these guys form a big globular colony. It's a really kind of soft, gelatinous stuff that you can pinch through. But for me, the clue is the color. I look for that beautiful grass green color or leaf green. That's going to show you that it's more likely to be a Phrydium than, say, a Phanathesis stagnina, which is going to have a darker, more olive green color, or sort of more the teal color. And then Mycologists have favorites, and this is one of mine. My absolute favorite cyanobacterial genus is Nostoc, and I have a license plate to show my love for it because that's what goofy scientists do. I'll show you some examples. This is Nostoc kilmanii. So if you have ever seen sort of these little mint green tapioca-sized balls or pearls floating at the margins of lakes, that is this particular species of Nostoc. So Nostoc kilmanii. I, it may have sort of a benthic stage, but tends to be more floating or planktonic. And usually it's about the size of tapioca pearls. Sometimes it can get to be a little bit bigger. So these are more like pumpkin seed diameter. So a little bit bigger, about 5 millimeters up to about a centimeter in diameter. But this is Nostoc kilmanii. I was sent this picture a few years ago of someone who wanted to know what it was. I think it's just beautiful. I wouldn't want it in my mouth. I wouldn't want it in my dog's mouth, but I think these guys are pretty cool. And the nice thing is you can um, tell that it's Nostoc kilmanii just because it has sort of this dull, dull light green color. This is going to have a really rubbery texture, so different from tapioca pearls texture. This is going to be pretty rubbery. You can press them between your fingers, but probably not break them apart by rolling them around in your fingers. So Nostoc kilmanii. Um, and then a little bit rarer are some of the larger species of nostoc. For me, these are indicators of really good water quality. These guys have to have absolutely clear water, and they tend to grow within aquatic plant communities that are really short. So things like quillworts or isaetids, you're not going to see these in lakes that have a lot of planktonic algae growing in the water because that would shade them. And you're usually not going to see them in lakes with a lot of large aquatic plants. So things like larger pondweeds, things like water milfoil, because that's going to shade them too. And these are actually, some of these species are an important part of things like Arctic ponds in the Arctic, and they're nitrogen fixers. This is an Arctic pond in Norway, just all of these great big rubbery colonies of Nostoc pruniforme, also called lake plums. This is what it looks like inside that colony. So there are strands of cells that kind of look like beads on a necklace that grow within a really rubbery matrix. So they have a very firm rubbery matrix. You absolutely cannot pinch them apart. You have to, I think this bit I had to actually cut with a scalpel blade out of a colony because it's just really super firm and rubbery. And this is a Nostoc pruniforme colony from a lake in Wisconsin. So these can get to be pretty big. At least in, I'd say, the Great Lakes region, I've seen them about 2 inches in diameter. So there's a special spring in the state of Oregon where they have what they're called mare's egg colonies, and these things are huge. They're like 6 inches in diameter. So someday that'll be my vacation to go there and see Nostoc pruniforme forming these mare's egg colonies in person. And then finally, this is a really rare one. So if you ever see it in a lake where you are, count yourself lucky. This is Nostoc zetterstedii, known as lake blackberries, and it forms in sort of these warty colonies that sort of look like berries. And again, this is something that indicates really good water quality. They have to have very clear water, very special, sensitive aquatic plants are what they coexist with. So these are really cool. These do grow in a few lakes in Wisconsin. So I guess let me know if you see these. I'd like to know where else they might be. And then I'm gonna finish up with some safety tips for pets. I'd say with a lot of benthic cyanobacteria, really the nice thing about them is when they're floating on the surface, they tend to be in big globs, and people can see them and they can avoid them, and they don't look like something that people would want in their mouth. But it's again like dogs aren't going to be that choosy about what they're seeing. And all of these recommendations come from cases that I've helped our Department of Health Services investigate where dogs and cats became seriously ill or unfortunately even died after exposure to cyanobacteria. So just always supervise pets when they're outside and keep them out of unsafe water. Prevent them from drinking untreated water and especially from eating floating material or material that's washed on shore. This is going to help them to avoid exposure to parasites, waterborne diseases, and cyanotoxins. And then when it comes to swimming for your pets, choose the clearest and cleanest water possible that you can for them and avoid shallow, warm, stagnant water bodies. That's where you might be more likely to have benthic mats of cyanide bacteria growing on the bottom. If a dog is wading, might kick them up, those mats up off the bottom, and then if it drinks the water, it might accidentally drink those mats along with the water. After they swim, always wash dogs off with clean water. That will remove any cyanobacteria from their fur, and that way they won't ingest it after they're licking themselves and grooming themselves after swimming. Um, Always provide access to shade and clean drinking water. Things like heat stroke can actually share symptoms of cyanotoxin poisoning, so it's important that dogs have a chance to cool down and always have access to clean drinking water. When it comes to like fetching in the water, use flat objects for retrieval. That helps dogs to close their mouth around that and keeps the amount of water they're ingesting lower. Keep splashing games really short and make sure your dog gets a lot of breaks from playing, especially if they're ingesting a lot of water. Water intoxication is another thing that affects dogs and can uh, the symptoms can be really similar to cyanotoxin poisoning as well. In some cases, things like really serious, like seizures and even death. So, if your dog drinks a lot of water, step in and keep them from drinking a lot of water. And then finally, if you live on a lake and you irrigate with lake water, don't use that lake water on your lawn during a bloom. You're basically bringing that bloom up onto the grass. And if your dog or if you even have a cat that likes to eat grass, they'll be exposed to cyanotoxins potentially that way. So these are all sort of recommendations for dogs that we unfortunately found out the hard way in Wisconsin. But I think really the good thing about these benthic cyanobacteria, I hope you have gotten some ideas of what they look like and just some of the things to watch out for in the lakes. And this is my contact info. And the organizers can share that with you after, possibly, and then I am happy to take questions. So I will stop sharing.
Great. Thank you so much. We are going to open this up to questions. You're welcome to either unmute and ask questions or put them in the chat, and I'm happy to read them out. We did have one question from Sandy about benthic in Vermont, which I'm happy to answer after this, but let's start with just some general benthic questions, and Sandy, I'll circle back to that one.
Okay.
Any questions? There's one in the chat that says, how long do you feel is sufficient to wait during the jar test?
How long to wait for the jar test? I think that kind of depends on what's in it. I have found that for planktonic blooms, it doesn't take too long. I've seen floating layers develop after 15 minutes or so. But when it comes to those benthic mats that you're shaking up and you're sitting near a window, I would give it several hours. Sometimes I'll just put it in the window and then I'll come back the next day. So let it sit overnight and then see what happens. Because I mean, while there are some cyanobacteria that are pretty fast at crawling around, a lot of those ones with like the more gliding type motion, it does take them a while to figure out, okay, that's where the sun is, and I'm going to go that way in the jar.
Right. We do have another question. What causes benthic cyanobacteria to come up to the surface is that first part of this, and is benthic worse than planktonic?
Okay, so first, what causes them to come to the surface? In many cases, if you have an area that doesn't get a lot of like wave disturbance or boat wake disturbance, it's just going to be oxygen that they're producing within the mats. It forms bubbles, and then they'll just kind of tear off the bottom and float to the top because of the oxygen bubbles in them. I will often see benthic mats kind of floating around near boat launches. So things like launching a boat that can kind of tear off the mats from the bottom and they'll end up floating on the surface. As far as what is worse, I guess it depends on if you are talking about, say, swimming in it. I think benthic mats are not as difficult to deal with. Let's put it that way. They're not as difficult to deal with if you're going to swim because, for the most part, if they're going to be a large mat, they're going to be noticeable. You can see them and then you can avoid them. Planktonic cyanobacteria, because they're like little tiny cells that are just throughout the water, they're a lot harder to avoid. So, hope that answers your question.
Great. I'm going to start with the next one, which is what is considered a benthic bloom? And are they ever considered to be dangerous to aquatic animals like freshwater sponges or other aquatic animals?
Yeah, well, I'd say bloom. There's no actual official definition in the cyanobacteria world for what is a bloom. I know you have in the ID cards that your volunteers have, you've kind of made a cut of, okay, this is just a little bit of stuff, and then this is a bloom, especially for planktonic cyanobacteria. But really, a bloom means there's an accumulation. For the most part, you can see it with the naked eye. So it might be there's cyanobacteria covering the entire bottom of a lake, and you might not see that unless you're snorkeling. It might just be stuff floating near the surface. It's kind of torn off the bottom, and then maybe it's just in a small area. So I know it's kind of a wishy-washy answer, but there's not actually a definition for what is a bloom. Um, as far as impacts on other organisms, I think Hannah Bonner is going to be speaking next in the series. She's done some really great work on stream benthic cyanobacteria, so I think she might be addressing some of that. And I think it's really an active area of research. We're most focused on impacts on humans, on pets, on livestock, because that's what we're most directly affected by. It's a lot harder to know how a lot of these cyanobacterial toxins impact other aquatic organisms. I'm not really sure about things like sponges. For benthic cyanobacterial mat forming filaments, it might depend on if they're starting to encroach on and grow on sponges. In that case, it might be more of like mechanical interference with them versus a toxin impact. So I think that's you would have to consider like what are the possible impacts could they have? Are they overgrowing something? Are they having toxin impacts that are directly affecting something? Are the toxins bioaccumulating and affecting higher levels of the food web? Um,
Right. Um, More specific question, when looking at potential cyanobacteria under a microscope, what features are most useful for separating oscillatoria type filaments from green filamentous algae or other harmless periphyton?
Sure. So the big signifier between cyanobacteria and other types of higher algae, so things that we're going to call eukaryotic algae, so things like green algae, um, are that those other types are going to have distinct organelles within their cell walls. So cyanobacteria are bacteria, which means that they don't have an organized nucleus, they don't have organelles. So planktonic cyanobacteria might have little dark things, speckles within their cells that are gas vesicles that they use for buoyancy, but that's not going to be the same as like having, say, a chloroplast, to define chloroplast that has green chlorophyll pigment in it, or a nucleus, or other organelles. So look for those organelles and the color too. So cyanobacteria, especially planktonic cyanobacteria, are going to look darker colored under a microscope, especially if they have those gas vesicles in them that diffract light and make them appear dark. But if you look at that ITRC guide, one of the appendices is a great big guide to cyanobacteria, and it has in addition to a lot of field pictures, it has microscope pictures too that should help you.
Right. The next question we have is how deep do benthic cyanobacteria typically grow?
That would benthic cyanobacteria, I think, are going to be limited by how deep the light can penetrate in a lake. So if it's a really clear lake, they might grow really deep. But if it's a lake that has, say, more phytoplankton growing in the water, that's going to create more shade. So they might be limited to more shallow areas of the lake. So it really depends on water clarity in the lake.
Okay. How well does visual abundance of benthic mats correlate with toxin risk? Are small mats sometimes high risk?
It would the the risk would all depend on whether that particular species within that mat are making toxins. Some species don't make toxins at all. Some might make toxins, but only under certain conditions. So the only way to know if there's an actual risk from the toxin is to do toxin testing on the mats. But I would say, like, when it comes to pets, I wouldn't want them exposed to any sort of mat abundance just because you don't know if toxins are present, and any mat material, because there's just thousands and thousands, if not millions, of cells within the mat. If they are making toxins, if there are toxins within those cells, that does present a high risk to anybody who's swallowing it.
Next question is, could you address how water temperature can impact blooms?
Sure. So as far as the planktonic or floating cyanobacteria, most of those species prefer warmer water temperatures. So that's why you typically see planktonic blooms in the summer, just when lake water is at its warmest. Benthic species, that might be different. There are some like those nostoc colonies, especially like in those polar areas, they might not be as well adapted for warmer water temperatures. So I think when it comes to benthics, there's going to be a spectrum of response. Some might like warm water temperatures, some might not do well just because they're not adapted for it. But in general, planktonic species of cyanobacteria that form like the most common bloom formers that we see in the United States, they really like warm water, which is why you see blooms most often in the summer.
So we do have some Vermont specific questions, which I can hold for the end, and I can also try to answer as well with our benthic monitoring program.
Mhm.
We do have one more that's, have you noticed consistent trends in the habitats where you're discovering benthic cyanobacteria, i.e. low, high wave action, protected areas, nutrient, high temps, etc.? And does this seem to be highly variable by taxa?
Um, yeah, like the person suggested, I think it does depend on the taxon. So for example, those planktonic mat, or excuse me, those planktothrix mats that I keep mentioning show up in clear water lakes in northern Wisconsin. I think they tend to grow more in sort of quiet, protected areas. Most of the examples where people have asked about them and sent me pictures, it looks like sort of quiet, protected areas that might have a lot of water lilies and pond lilies, for example. I don't think there's like a lot of wave action, which kind of probably helps those mats to stay developing on the bottom. Something I don't think you would have a lot of benthic species necessarily developing on a really wave-washed shoreline. So a lot of these guys are going to want more protected areas. I mean, granted, if you're one of those motile species, you can still crawl around and reform a mat, but that's sort of energetically expensive. So you are probably going to see a lot of these benthic mats more in protected areas to just kind of give it a quiet, calm place to grow on the bottom.
Great. Thank you. Um, so the one question that we skipped past earlier is, when did we first cite benthic in Vermont and how frequent are the sightings? I know that Nostoc actually was first observed in 2019 twice in Lake Winona, and there have been several reports since 2022 of different benthic species on Lake Champlain and Lake Maury. However, that does not mean that they have not been there. This is just that someone had noticed them, and those were the sightings that occurred. Um, we did launch a program last year in collaboration with Tetra Tech, where we are looking to understand the extent and the geographic distribution of benthic cyanobacteria around Lake Champlain Basin specifically, and get a better understanding about how frequent those sightings are, because the more people know how to recognize them, the more we're going to be able to understand where they are growing and when they are kind of popping up. And so, Geraldine, to answer your question about what benthic species appears only in late fall in Vermont, uh, we are really not sure. Would love to know more about that. And as we kind of continue to learn more and collect more data, um, we will be publishing on that, and you can read up, and happy to do another webinar series talking about our results. Are there any other questions for Gina? You're either welcome to unmute or put them in the chat. All right, I am going to stop the recording and thank you all for joining us. We really appreciate it. We will be returning with a webinar series again next Tuesday, so please join us. If you did not sign up for that, please just let me know. I'm happy to put you on the email list. It is the same Zoom link, so you can also just use this link as well. Thank you all.